Membrane transport. So why does this make any difference in human nutrition? Why deal with membrane cell biology? Well, the reason for that one, everything you eat has to pass through a membrane for your cells to be able to get it. So we need to look into that a little more in this video. When we start talking about membrane transport, one of the main things we look at is the plasma membrane. With that being said, how do we get ions, molecules, electrolytes, water, everything across the plasma membrane? Because every one of the cells in our body, from the cells of our intestines, to the cells in our bones, to our blood cells, to our nervous cells, everything has a plasma membrane. And that plasma membrane is made up of a bilayer of phospholipids. Now, the phospholipid bilayer is going to end up having two phospholipids, basically tail to tail. Reason being, the tails of the phospholipids are hydrophobic. Hydro water, phobic fearing. So they're water fearing, so they face in. While the head groups, the ones that are facing out, are hydrophilic. That means they're water loving. They have no problem touching the aqueous solutions inside and outside of a cell. So the inside cell and the outside cell is full of water, full of ions, full of all these different components that we have to regulate. Now, water can travel through the plasma membrane pretty easily because of the things called aquaporins. Aquaporins are protein channels that allow water to travel from a high concentration to a low concentration of water. Just travels down its gradient. Good old passive diffusion. Now, there are other proteins that will help to control how other ions and other molecules will move. There's one we'll look at called a cotransport. In the case of a cotransport, it's one molecule is moving and it kind of pulls the second one along for the ride. So really the energy or the gradient is built on that first molecule, that first ion, while the second one coming along is kind of tagging on. It's not really on a gradient, not really there. It's just, hey, this is working, I'll be pushed on through as well. We'll see that a lot of times with sodium, for example, where sodium travels through down its concentration gradient, and then another molecule is being pulled along for the ride. When we start talking about sodium, potassium, chloride, proteins, all these cations and anions, so cations positive, anions negative, they're kept at different sides of the plasma membrane. Now, this is set up to help with various different body functions. So it's kind of interesting because most things in the body are trying to maintain homeostasis, an equal balance. But the plasma membrane is trying to keep the ions and molecules out of balance. Because the sodium will be kept in a higher concentration on the outside of the cell. Potassium will be kept at a higher concentration inside the cell. This helps to set up a gradient across the cell. By setting up this gradient, you put energy into it to make the gradient because the molecules don't want to go into a higher concentration. So by using what's called the sodium-potassium ATPase, or sodium-potassium pump, you're going to allow ATP, energy, to move 3 sodium molecules and 2 potassium molecules. And you build this high concentration of sodium outside the cell with a low concentration of sodium inside the cell. So when the channel is open and sodium can move, it travels high to low. The concentration gradient is set there. The gradient has stored energy. Well, if you think about stored energy, think Think about a waterfall. The water up top has that potential, that stored energy. When released, it flows down to the bottom, releasing that energy as it goes. That's why waterfalls, or running water for that matter, could be used to power different mechanisms. So we're setting up these gradients based on the sodium-potassium pump, based on co-transport channels, based on water concentrations, aquaporins, all trying to set up our cell's homeostatic state, which in this case means we're setting up concentration gradients. Now, there's another part to it as well, looking at tonicity. Tonicity is looking at the dissolved particles in a solution. So we have three different names here. We have hypertonic, isotonic, and hypotonic. So hypertonic. Hypertonic is a high concentration of dissolved solutes. Hypotonic, so hypotonic, is a low concentration of dissolved particles. While isotonic, ISO means even, so isotonic is referring to a balance, neutral. You want your blood to be isotonic. You want any solutions going into your blood to be isotonic. Your body tries to maintain an isotonic environment. That's why if you ever have an IV bag a lot of times, and you're not trying to increase or decrease hydration, you're trying to maintain, that IV bag, besides the water, will have salt that makes sure it's isotonic to your cells. Because you want to maintain the level, not change it if everything's okay with the hydration level. Now that hypertonic, so hypertonic, means you have a high amount of dissolved particles. If you take your blood and you have a blood cell in a hypertonic solution, that means the solute around this blood cell will have a higher solute concentration, more particles. That means there's higher particles out here that can't get in, lower particles inside that red blood cell. The only thing that can move is water. So what happens is the water is going to try and leave the red blood cell and try to dilute the external environment. That means that red blood cell is going to shrivel up, not be normally functional. So in a hypertonic solution where there's too many particles dissolved, cells lose water to try and reach a homeostatic balance. Because the ultimate goal is to try and balance those ions from inside to outside those particles. If you're saying, well, hold on, didn't we just say that sodium is kept higher and potassium is kept higher? I mean, we had talked about that, right? That's looking at muscle cells, neurons, only certain cases you try to maintain there. Not everything is trying to maintain a gradient. There's plenty of other particles that are in your blood that want to be in equal proportions on both sides. So if you have those particles in high concentration, you can create a hypertonic or hypotonic solution. And it can still happen with salt, sodium, chloride, potassium, because even if they are having a gradient, there can still be too much of a gradient, too much outside where the cell tries to balance. There's limits to everything. So let's try an example of this. Let's say you have a blood cell, which is balanced properly, it's isotonic, but it's put into a hypotonic solution. Hypotonic, low solutes around the cell. What happens in that blood cell? Well, if you're thinking, okay, the cell is normal, what's around it is a low concentration, water's going to go into the cell because that's where the higher concentration of particles are. The cell will swell up, potentially burst. So therefore, that means If anything's put into a hypertonic solution, it shrivels. If the cell is put into a hypotonic solution, it swells. If anything's added to an isotonic, everything's happy. It stays the same. The whole idea here is water follows the solutes. Water follows salt, for example. Because water can move freely, but that semipermeable membrane doesn't allow the different particles to travel back and forth. So water moves to try and reach an equal concentration of water on both sides. Membrane transport is kind of important for how we move particles. And as far as nutrition goes, we have to know this because that's how we get those things from our intestines that we ate into our cells and make sure our cells can work. Until the next video.